Hi, my topic is going to be on aldehyde dehydrogenase 2 and a variant allele ALDH2 star 2. So the reason I chose this topic is that I am personally affected by this, and I was just having a drink one night and I had an unusual bad headache afterward. So I was just wondering about the alcohol metabolism, and that leads me to this. Um, so it touches on many topics that we've learned from 255. It's got something to do with metabolism and some enzymatic reactions. We will even see some glycolysis later on in the primary article, so it's pretty fascinating. So what most people know about this allele is its relation to alcohol metabolism. So I myself have this variant. If I have one drink, my face sometimes could turn really red, and this is what most people noted as the Asian flushing. It is because when we drink alcohol, the ethanol in alcohol will get broken down into acetaldehyde by alcohol dehydrogenase, and then the acetaldehyde will get broken down into acetate by aldehyde dehydrogenase 2. So aldehyde and acetaldehydes are harmful to our bodies. It impairs cellular functions, and frequent exposure has been linked to higher risk of cancers and cardiovascular disease. When we have the wild type, the acetaldehyde get broken down into acetate pretty quickly, so that wouldn't be an issue. However, with the variant, this is where the problem comes in. So aldehyde dehydrogenase 2 is only one of the many aldehyde dehydrogenase family, but it's been the most effective in metabolizing aldehyde in our system. So that's what it's been focusing on, and ALDH2 and the ALDH2 star 2 variant has been found since the 80s, but in the early days, the focus have been on its relation to the alcohol metabolism and for it being a risk factor for esophageal cancer. But recent studies also suggest that the ALDH2 dysfunction may also be associated with a higher risk for osteoporosis, cardiovascular disease, and some other neurodegenerative diseases like Alzheimer's disease. So ALDH2 star 2, this allele is found in about 35 to 45% of East Asian descendants. It is estimating that 560 million individuals are being affected by it. Obviously, it is more prevalent in Asian countries. It could go from 28% in Korea to as high as 45% in Taiwan. But obviously, it is also affecting Asian American populations. Aldehyde is present in a lot of different things. It could be found in food, beverages, fragrance, so it could be found in our environment. So my article talks about alcohol dehydrogenase 2 and its deficiency of it is leading up to increased oxidative stress and how the body tries to deal with the extra oxidative stress by remodeling the metabolic pathway. So I thought it's really interesting, and it really touches on topic beyond just the alcohol drinking. So I want to share this article on the presentation. So before we get into more on the article, let's talk about a general aldehyde dehydrogenase mechanism. So this is how the enzyme metabolize an aldehyde into carboxylic acid. So we have an active site that has a cysteine thiolate and a glutamate. And because of the negative charge glutamate, we have a proton transfer, and that activates a cysteine thiolate. And then so the cysteine thiolate will then launch a nucleophilic attack on the carbonyl carbon and form a tetrahedral intermediate that is going to collapse. So the hydride that is being removed will reduce a NADH. Will reduce a NAD plus to make it a NADH. And after that, we will have a trigonal planar complex, and there will be a hydrolysis step. So the glutamate in the active site will deprotonate a water, and then it will attack the carbonyl carbon on the thioester bond. And now we have another tetrahedral intermediate, and then the sulfur will get kicked off as a leaving group. And there we'll have our carboxylic acid. So the ALDH2 allele is a mutation that is caused by a single point mutations that went from G to A and that changed a glutamate to lysine. What that means is that the mutations will have a NAD plus binding site that is altered, and the catalytic activity is lost because the physiological levels of the NAD plus just cannot meet the demand of the ALDH2. What that means is that it will leave us a 200-fold increase in CHEM for the NAD, and typically the ALDH2 carriers have a lower than 50% of the wild type enzymatic activities. In the study, they expressed the ALDH2 genes in mice and compare it to another one with the wild type ALDH2, and they look at different measurements. They look at the body weight, skeletal muscles, and different cardiovascular performance, and they found that the mice with the ALDH2 genes was strongly expressed in the cardiac and skeletal muscle. And then they also found that the mice is expressing phenotypes such as smaller body weight and less skeletal muscle. And they do not think that should be attributing to the ALDH2 activities. So they are suspecting that it may also have something to do with the ALDH subfamilies. And in addition, they also find that there is more oxidative state associated with the mutated genes on the mitochondrial redox state. So this part of the study gets really interesting. They did an echo exam on the mice, and then they find that the mutated mice actually have a normal ventricular systolic and diastolic functions, indicating that the heart somehow found a way to adapt to the increased oxidative stress. So they did transcriptome analysis on them as well, and then they find that there's increased glutathione expressions that they see in the mutated mice, and they also find that there are more pentophosphate pathway that is being taken during the glycolysis to get more NADPH generated, because that's what required in the glutathione redox cycle. And glutathione is an antioxidant, and that could be explaining how the cell is dealing with the increased oxidative stress. So this study shows that this ALDH2 STAR2 allele really have a huge implications that goes beyond just the alcohol metabolism, and I just thought that is really interesting. So my questions for the discussions would be, what do we think that could be responsible for the phenotype differences that we see in the mutated mice in terms of their body size and the muscle mass?